This is an ancient and a modern fermenter. So people have been fermenting food and drink for thousands of years. And so the Neolithic jar on the left, and then the full-scale fermenter. There, the microbes are doing the same thing in that Neolithic jar versus that fermenter system on the right. We'll talk a little bit at the end of the chapter about the enzymes that microbes produce, and a lot of companies that are marketing these biological products in biotechnology. This is a new section in your third edition that I really liked. It's about a compound called alginate. And alginate is typically harvested from this Pacific giant kelp that's shown on the left, and has been in use for hundreds of years because this is where we get agarose. And agarose is basically this alginate compound, and if you'll remember back in the beginning of the course when we talked about Mrs. Hess contributing in Dr. Koch's lab this solidifying agent. So the alginate has been used to solidify jellies for hundreds of years, and so that was a real contribution that Mrs. Hess made in Koch's lab to solidify media into the agar plate. So, of course, this compound is near and dear to us microbiologists' heart. But like I said, it's been used in food preparation, jellies, and so on. And so on the left is this great picture. A number of you who have been to the Pacific, I'm sure, recognize that kelp, and then it's part of why the Pacific smells so great. It's a great ocean smell is from that kelp. And so on the right, this compound is produced naturally by a number of species of Pseudomonas. Pseudomonas aeruginosa produces this stuff in our lungs and gives us cystic fibrosis. It didn't mention that. And that's in the people that have this genetic defect that they cannot clear an infection. Pseudomonas aeruginosa, which is shown on the right, produces the same alginate compound and builds these biofilms. Has been engineered by a company to not be pathogenic and simply pump out pure alginate, which is much easier to package and safe and so on. So a couple times through the chapter we'll be looking at some biotechnology, and here's a nice example of you can get bacteria to produce it. It's going to be a lot easier to manufacture it. And here's some alginate put to a different use. So it says modeling cast from microbes. And so this is in the production of prostheses. And so here is a cast that is made from alginate. And then you can take a mold of people's digits or arms or whatever, and then to make these kind of plastic-like prostheses. And so just another neat use of this really interesting natural plastic called alginate. So it said, excuse me, it was the first biotech startup company in West Virginia is marketing this bacterial-produced alginate compound. So alginate is a good thing to focus on in this chapter. There's other chapters that we're just going to be looking at pictures of food. And that'll be neat, but mostly for your information. This looks like for your information. Now, Agaricus bisporus, I haven't tested on that in the past. It's the button mushroom that is marketed, in the grocery stores. And isn't that portobellas? P-O-R-T-A, portobellos. And they all typically grow on some sort of horse manure. And so, good idea to cut off those ends before we fry them up, saute them up. But that has not, I've never heard of mushrooms being a source of food poisoning, except the ones that are toxic to you and will kill you in about 3 minutes. That's a pretty harsh form of food poisoning. Underground truffles, incredible. They're very, very tasty and very, very expensive. And I bought some at a grocery store, about 1 ounce for about €4 and put it in some spaghetti. It's great. Okay, here's a little bit of detail here that is worthwhile. The top part of this is we've talked about this red algae porphyra, porphyra, and then the compound, the carbohydrate, porphyrin, is the kind of the main structural component of the seaweed that is used in sushi to wrap the rice and raw fish and so on. And so just harken back to chapter 13, when we talked about the interesting study of the Japanese group that has found the enzymes, the genes that come to break this down, most likely came from marine bacteria. So different people groups that have been eating sushi for thousands of years now have the capability of breaking down that porphyrin carbohydrate that is in that seaweed that's used in sushi. This section also talks about consuming, and that's what we're doing. We're talking about consuming microbes directly, so consuming mushrooms, consuming the seaweed. These are red algae in the case of the sushi. But then there's also this organism called cyanobacterium. So there's been an emphasis for a long time to make single-celled protein. Just grow up bacteria, harvest them into cakes, and eat them as an efficient source of protein. And it's worked out to some extent. One of the problems is the nucleic acid content in bacteria is too high, and it gives us kidney stones because we convert these purines to uric acid, which precipitates and hence the kidney stone. So the cyanobacterium, which is pictured here on the right, spirulina, has lower purine content, and so we can eat more. And you can see this in health food stores. Spirulina. Anybody see this around? Anybody eat spirulina? It's kind of a
green.
Pardon me?
It's like green.
Yeah, pretty bright green. Yeah. It goes in and out of, but I've seen people eating it in the last few years. So great source of protein and no problem with kidney stones. Okay, so food fermentation. I've been on a kick in the last few years to I've been making sauerkraut, my wife's been making yogurt, and just that process of fermentation is definitely a value-added system in terms of adding nutrients, and a great source of probiotics. So as you ferment the food, you're growing up these populations of healthy bacteria that are healthy for your GI tract. So it's win-win. Preserves foods, so some of the purposes, traditionally you acidify these foods, and so they're preserved. You can preserve them for a long time. And then this is kind of what I'm talking about, to improve digestibility, breaking down fibers, but also this better insulin response. So in the fermentation process, the organisms are taking those easy sugars, which give us a fast, unhealthy insulin response, and breaking them down to fermentation products, which don't give us those bad glycemic responses. And so there's healthier metabolites. And then they add nutrients, vitamins, lactic acid, and flavor molecules. So those fermented foods and these really interesting these compounds produced in small quantities that really add to the flavor. And sure, they typically, if you don't boil it afterwards, if you eat it, in a way that preserves the organisms, they are a great source of lactic acid bacteria, which are generally really good for us. So what's interesting about all these fermentations is it doesn't take a PhD microbiologist to do this, right? The more you know, and the more you try to mess with it, the worse you mess it up. So the point is, what is naturally on that cabbage is going to ferment that cabbage into sauerkraut. And so you don't typically inoculate any of these fermentations. They are found as indigenous flora. So the organisms, the microflora, are indigenous, are native to the material that you are fermenting. Sometimes starter cultures are needed but typically that's a passage from a previous fermentation. Okay, this I end up saying this is going to be for your information. I'm not looking at any details here, but there are acid fermentations that produce acid, which are pretty common, and then less common are these basic or alkaline fermentations that produce ammonia. So two routes, the acid and the alkaline. So a little bit about dairy fermentation, especially just making cheeses. Lactobacillus, streptococcus are the two genera that are involved in that. And if you've ever done this before, all you really have to do is acidify milk, and it will then precipitate. So it's this interesting effect of the pH drop causing the casein protein in milk to lose its three-dimensional structure, its native structure, and then just fall out of solution and coagulates into a solid. So then these peptides coagulate to form a semi-solid curd, and then the liquid portion above that precipitate is the whey, and full of casein protein. I don't know, this is sort of, for your interest. The unripened cheese, these fresh cheeses, you can make them overnight and they're ready to eat the next morning. They're really great. Goat cheese, in an omelet can hardly be beat. But then it's with time that the hardening process happens, where you lose water and then you gain flavor. So it's almost, for one thing that's going on is it's concentrating as it's ripening, so it becomes harder, you lose water, those flavors concentrate, but then there's also these other flavors that are produced. Feta cheese, also great. That's typically made from sheep, which I'm remembering, I'm just going to show some pictures of our sheep today, because we had the big exchange. I have a guy that I've gotten to know who raises a number of sheep, different types of sheep, and we decided to do a swap. He was going to take our ewe, which has been producing a lot of lambs over the years, and he was going to give us his young studly ram. And so, on the very day that these poor girls lost their mother, and it was very traumatic for them, I showed up 1 hour later with Mr. Ram, meaning Mr. Right, and everything was okay. They got over it. They forgot their mother once they laid eyes on this guy, who now is the maestro of the property. And I am on my best behavior, because with previous rams I have misbehaved and wrestled him because he always rams me. And now I carry a stick behind me, And just to keep them from doing that, because mixing it up with these big rams has been a bad deal. There was a class, somebody in this class that took care of me in the ER that one night, who was helped stitch me up, and so now I'm on better behavior. But like I said, there were supposed to be pictures of sheep in the beginning of this lecture. Makes me wonder which version I'm working on. Okay, yeah, I mean, this is spring lambing. There's all sorts of lamb stories this time of year, so I'll bring in more stories. Okay, cheese production. This is for your interest. This is, I'm not going to be testing on how you make cheese in other classes. When we actually do this, in my environmental microbiology class, we have a laboratory, and we made yogurt, and we made sauerkraut, and we made kefir. And hardly anybody wanted to taste it afterwards. I was the only one. I took them all home and guzzled them. I have some pictures of me guzzling something out of a test tube. It looks pretty rough, but it is, it was yogurt. One of the interesting things is solid curd is cut. This is where the word cheddar comes from, is a certain cut of that solid curd. And then you press the curd into a mold, and then the cheese is ripened or aged. This is especially true with those hard cheeses. This is pretty much for your information. Microbial metabolism generates these byproducts that confer this characteristic aroma and flavor. This is a billion-dollar industry because we crave these small concentrations of methanolthiols and amines and thioesters and aldehydes. It's these compounds that are produced in fairly small quantities because they are potent to make distinctive cheeses. And so this is certainly a science in and of itself. Okay, so how about moving on to vegetable fermentations? A lot of these tempeh and what do we typically eat? What's the white curd that's in the grocery store from soybeans? Not tempeh. Tofu, thank you. Yes. So both of these are made from beans, and so high protein, and yet those proteins are broken down, and so it's more easy to digest. Rhizopus is actually the beans that are permeated with mold. So you're both eating the microbe itself as well as the fermented product. Yeah, I put this in last year. Just a note to all the guys in the room, there's a number of interesting compounds in soybeans that mimic female hormones. So these estrogen mimicking compounds, they just structurally are very similar to mammalian female estrogens. And so they're about 1,000 times less potent, but hey, hormones are really, really potent. And so that's one of the interesting things about soybeans. Who would have thought that compounds in plants mimic mammalian hormones? Okay, and then there's the whole industry of kimchi, which is then the Korean version of sauerkraut with a kick. So they take the German sauerkraut, and of course they're using different cabbages and spicing it up with that red pepper and making it delicious. Lucana stock. So how about these two names? I've Rhizopus to make the tempeh, and Lucana stock to make sauerkraut, pickles, and kimchi. Those are two genera that are worth remembering. Some representative material from this chapter is what I'm going to try to give you that heads up. Okay, this is an important part of the chapter because it's the most interesting. So we're all interested in chocolate. And in this version, they've got this marvelous picture. I did not know that the coffee bean came from such a gorgeous pod, this bright red pod from which produces the cocoa bean. So inside this pod are the beans, but they're completely surrounded by this mucilaginous material that is key to making chocolate. So if you take that cocoa bean and ground it up, it's absolutely nothing like chocolate. You can't just extract these beans. It has to be fermented, and it is a completely complex fermentation that nobody even understands yet. And so a number of microbiologists, and your text highlights a Brazilian woman, a microbiologist who has been getting some understanding of the fermentation process, but even still, you cannot do this in a factory. This is all done in situ in the countries where they produce the cacao bean, in Brazil, South America, and Africa. And so it is from the tree Theobroma cacao, and Theobroma means something like drink of the gods. Broma, something to drink, and God. And so, for thousands of years, people have been kind of overseeing this fermentation process, helping it along, and making chocolate. And so it's this a very complex combination of native microbes that are on the leaves of the cocoa plant, and we'll kind of expand on that in this outline of what happens right here. So 3 stages. So it occurs within this fruit pulp. So we're talking about this really heavy skin. For one thing, it's really hard to get that skin off of that cocoa bean. So letting microbes digest it and acidify it releases the actual bean itself. So there's many reasons to let nature do its thing, and that's just one of them. So I'm kind of giving more detail on this first step, but there's basically 3 steps that this microbiologist has given us to help us get some understanding of the process. And this is carried out by yeast, and the two genera of yeast is Candida and Saccharomyces. So these should be italicized, genus names. And it's an anaerobic fermentation, and this high pectin pulp. So there's a lot of this complex carbohydrate called pectin, which I'm sure you've heard of, is dominating and clinging to these beans, and this is what is fermented by the 2 yeast, Candida and Saccharomyces. And that long chain carbohydrate is converted to glucose, and the whole system liquefies, which on the surface, a couple of these stages of fermentation, my brother has done some site visits of these cocoa locations, trying to kind of stimulate indigenous economies. And he says some of this, he said, you would not recognize the chocolate. It is disgusting. Some of these steps of fermentation make, fumes, and it's unrecognizable, and you'd never think it's chocolate. But with a little patience and a couple more steps, it does get to be the drink of the gods. Okay, so specifically what's interesting right here, ethanol is produced in that first anaerobic fermentation step, and the ethanol actually breaks down plant cell membranes, which release plant enzymes, which then are part of producing this unique chemistry of the chocolate. And so this is why chocolate is addictive, is because there's a few secondary metabolites that are produced that are stimulating, and so there's actually even antidepressant neurotransmitters produced. And the irony is this is actually plant enzymes that are now participating. So you've got bacteria, you've got yeast, and you've got plant enzymes all participating in this. This is why it's so complex, process of fermentation. And has yet to be identified. Not all the steps and not all the enzymes are known, because it just can't be made in a factory. It's all kind of indigenous industry. Okay, so that is some details on that first few steps. And kind of interesting that the alcohol just breaks down plant cell wall, just releasing these plant enzymes, which then go to making some of the key flavor compounds of chocolate. And then after that, lactic acid bacteria take over, still anaerobically fermenting. And then there is a aerobic respiration carried out by acetobacter. And so there's some turning over of the material, as far as the people who are overseeing this process, which then allows for oxidation. And then that gets rid of the ethanol, because this drink, we don't want ethanol in this, in the chocolate. So on the left, a lot of times they use plantain leaves, these big leaves, to carry out the fermentation. So this is like a third of the way through of the process. It had been covered up by these plantain leaves. And so, like I said, it's the plantain leaves, or whatever leaves that are being used in that indigenous country, that contribute enzymes to the process. Really interesting. So, incredibly complex plant, meaning plantain leaves and cocoa bean. Bacterial, and that should say yeast. And this says microbial enzymes. Who knows what that says? Mix, yeast mix of enzymes. Do you have that on your outline? And you can read it? Yeast mix of enzymes. So that's what's being shown on the left. And understand, this is not going on in a Nestle factory somewhere. I always thought Nestle is, Switzerland makes chocolate. No, Switzerland doesn't make any chocolate. The chocolate is being made in Africa and South America. And it's imported to Swiss, and then they add some Swiss magic. I do understand the Swiss and Belgian chocolate magic that they add, but the chocolate's actually made, has to be made, not in a factory, out in the field. And so that's one reason that's a neat picture. It just shows you this is where it's happening. Okay, so that's on the left, and then here are 3 lines. So this is the progression and succession of natural microflora, ebbing and flowing over a 3 or 5 day period of time. And so then over on your y-axis is the log number of cells. So here you've got 1,000,000 cells, and up here at 10 to the 9th you have 1,000,000,000 cells. And so here is this is some of the data that I don't know her name, the Brazilian microbiologist has acquired. That initially, here's the population of the yeasts that are increasing and making the ethanol, and then their populations dive, and then the lactobacillus, second step, anaerobically ferments and changes those sugars to lactate and acetate, and then lastly, the acetobacter in red shows the population increase of those organisms, and what they're doing is then oxidizing the ethanol. And so that is in response to this turning of the beans to make it aerobic. So production of chocolate still largely undefined, has to occur at site of harvest, in situ in those countries. So really interesting process that's still not near completely understood, and it's a process near and dear to our hearts, right? We all love chocolate. And it's all microbes that are doing the magic. Well, it's all natural. I mean, so it's not just microbes, it's the plant enzymes as well as bacterial and yeast mix. Any questions on chocolate fermentation and production? Okay. So moving on to alkaline fermentation. Natto is is this beans that have been fermented, and this sticky material is this extracellular kind of polysaccharide and is the microbe themselves. And so, a lot of these fermented foods, a big influx of healthy probiotics. 50,000 tons of natto are consumed per year in Japan. And then some of these are, pretty darn cultural. I don't know if I would do this one. This is a, they call them 1,000-year-old eggs because they've been fermented, and so the white is now black and the yellow is now green, and these are pretty potent. If you can imagine, anybody had one of these pidan eggs? Me neither. And that, I may not go there, but.
So where would you have like certain nutritional value, or is there?
Well, you can store them for 1,000 years. I mean, there's one thing. And then it's kind of an adapted taste, an acquired taste. At some point, you just say, this is the kind of egg I want this morning. But certainly you got the preservative working. Coagulated by base, sodium hydroxide, fermented by bacillus, and green eggs, and what do you have that with? We have green eggs at home, okay? But these are green eggs laid by Araucana chickens. And so they're on the outside, kind of a nice turquoise green, but on the inside they look normal. Okay, so moving on to bread and beer and wine fermentation.
Can you go back?
Coagulated by sodium hydroxide, fermented by bacillus. And that's Dr. Seuss at the bottom. We good? I'll take a swig of my peppermint lane herbal tea. I started drinking coffee on Tuesdays and Thursdays. And I've been getting a lot of work done. I get a lot of work done on Tuesdays and Thursdays. And on Monday, Wednesday, Friday, it's peppermint tea for me. Okay. So one point here, I'm beginning to concern about my, this has gotten a bunch of new stuff. Anyway, this version. Saccharomyces cerevisiae. This is an important genus species because not only is it baker's yeast, it is also brewer's yeast. It's the same thing. This is the same organism that carries out fermentation for bread and for wine and beer because they both are producing ethanol and carbon dioxide. So the CO2 is then what causes the bread to rise. And then when you bake it, the ethanol is driven off from the bread. Easier to chew and digest. That's one of the roles of carbon dioxide is to make it lighter. And then there's these other breads that are pretty interesting. I've had one of these, and probably not going to test on this, but anybody had this Ethiopian bread? It's made from teff, which is a grain which does not have gluten. And there's these huge sheets. It's like a tablecloth, real thin, and you can fold it. And I had this at a Ethiopian restaurant in DC one time. Anybody had this this injera Ethiopian bread? It's nice. It's tasty. And so, interesting. Candida is a genus that can be pathogenic, but understand Candida can also have species that are really important. And this is the second time we referred to it. This genus can also be helpful and involved in food fermentations. Okay, Saccharomyces cerevisiae, baker's yeast equals brewer's yeast. So one of the things about, let's see, what's one of the words? The barley, I forgot the term. Yeah, malted barley, which has always appealed to me because I love chocolate malts, and I always ask for more malt, and I'm sort of addicted to whatever that flavored malt is. But anyway, malted barley is simply the barley seed that has been germinated. And so it's a little seedling, it's a little sprout of barley that then is used in the fermentation. So that's what malted means, is it's just been germinated, and it's the germinated barley grain that then make these enzymes to break down the starch, then allowing for the yeast to take over. So, you know, a lot of these fermentations you can see is this interchange between plant, fungus, and bacteria to make the fermentation products. And then it's these esters that make, you know, the IPA that we know and love, and all these wonderful flavors in beers. So this slide looks an awful lot like this slide. But in terms of making wine, again, it's the same genus species involved making bread, beer, and wine, Cerevisiae. And one of the interesting points is the difference between white wine and red wine. That ignoramuses like me said, oh yeah, they just use the white grape or the green grape and they use the red grape. But they use the same colored grape, typically red, and then it's with skin to make red wine and without skin to make white wine. At least that's a big group of red and white wine. So the skin is removed for white wine, but not for red wine. And then we all know it converts the sugar to alcohol. But then this malolactic fermentation by this genus of bacteria. And so realize that bacteria also participate in reducing the acidity. And it's to get rid of this malolactic acid, this Onychus onii genus. And that is showing, well, two things are shown here. Just where in the process do you remove the skins? And look at sulfur dioxide is just added, you know. So how can we get rid of sulfites? Well, you know, talk to the winemakers who just add it first thing. You know, sulfites aren't necessarily the best thing for you, but it is just totally part of the process. So I say note role of the bacterium Onychococcus oni. Oh, I was going to say onii, but it's not really onii, it's oni. In this secondary fermentation to reduce the acidity by removing malic acid. So here's this malolactic fermentation, which is really what I should have asterisked, which is what is needed to reduce the acidity in both red and white wines. And so same colored grape, skins removed early versus later with red wine. Then it's those skins that give a lot of those tannins and stronger flavors in red wines that make it distinctive. And then you know, release those. I'm trying to remember the name of those. Are those carotenoids that give you the red color? There's a developing wine program here on campus in the College of Ag. Has anybody taken any of those courses in the College of Ag? They've got it's a nice program. So interested in winemaking, there are courses in the College of Ag that are offered. Okay, a few more minutes. Putrefaction versus rancidity. I'm not going to cover this, but can you understand putrefaction when you look at the name of the chemicals called cadaverine and putrescine? Do you think those are going to knock you out? Yeah, they are these amine-containing molecules that just are, you know, the essence of the cadaver, the essence of putrefaction. So we definitely have the repulsion at almost all of our sensory levels to not eat that. Okay, food spoilage, soured by excessive fermentation, made bitter by proteolysis. So it's these amines which make those odors, which I just gave you the two examples. One of the interesting things right here is how seafoods spoil rapidly because they've got these unsaturated fatty acids that quickly oxidize. And these psychotrophic bacteria. Psychotrophic means organisms that have the ability to grow when it's cold. So they are growing in a refrigerator, for example. And so a lot of the bacteria that are on fish grow in cold water. And so they are still on the fish when they are on ice, and they're still metabolizing. And that fishy smell is the reduction of TMAO to this trimethylamine. And so often, when you say, oh, I don't like fish because it tastes fishy, well, that's because the fish is not fresh. And you're tasting this trimethylamine. And so give fresh fish, like go out and catch it yourself. And then if you have that opportunity, grill it, and you will not have that fishy smell or taste. Okay, I'm going to finish up with a couple of things, and that is some outbreaks of food poisoning, and this is salmonella in peanut butter. So even here in Las Cruces about 6 or 8 years ago, and across the country, there was a huge recall of peanut butter because Salmonella enterica was growing and then causing, this is the cases of food poisoning from October to January, spanning the year 2008 to 2009, that were breaking out in a particular state, or maybe this is countrywide. And so there was almost 700 people that became ill from peanut butter. And one of the interesting things here is the nice DNA forensics that were done and are typically done in a lot of these food poisonings, where you can extract the DNA and fingerprint the causative agent of the disease. So this is Robert Koch's lab 100 years later using DNA techniques. And so humans with the illness and this particular jar of peanut butter matched up. So here's the DNA fingerprint of somebody sick, and here is the source of that illness, and this is Koch's postulates. This is what they are doing in modern laboratories, is proving cause and effect. What is the causative agent of the disease, but using DNA fingerprinting. And then here is a DNA fingerprint of another organism, but that wasn't causing the disease. So kind of a nice example of Koch's postulates being carried out in 2008. Okay, this is for your information, but I only touch on it again because we've talked about pathogenicity islands. So the factors that contribute to food poisoning are often encoded together on these groups of genes that are called pathogenicity islands. And so here's a pathogenicity island from the salmonella that caused the food outbreak. Okay, I got a few more minutes to finish this off, I think. Industrial microbiology is the exploitation of organisms, and we're going to be talking about production of pharmaceuticals and enzymes are the two things I'd like to focus on. And generally, this idea of bioprospecting is now a type of bioinformatics. So searching the genomes for commercial products, for genes that will code for enzymes that can be used in commercial applications. And so what are the traits that that these microbes or sources of enzymes have to be? They have to be genetically stable, inexpensive to grow. If it was a pathogen, you better knock out those pathogenicity islands and be able to grow a safe product. If you're interested in that enzyme production, how can you optimize for high level of protein? And then how can you harvest those proteins? We have patented a polymer that a yeast produces, and we're doing this stuff in my laboratory right now. Thomas Behill is the student that's working on this polymer that we've got produced from a yeast. Okay, so I will cover this at the beginning of next period, but this is a fun slide that we can finish up with on